Okay, so now we have the second part of the lecture, and we are going to talk about more practical part, which is the agarose gel electrophoresis. So first, of course, we want to load our gels before we run it. So what we do, we actually fill the tray with the buffer, and you know the running buffer is TAE, it is 1X. It should be covering the gel and should fill the wells of the gel. So if you look from here on the gel, you will see something like this. So these wells are the places that we are going to load our samples to. So the level of your TAE should be above it, so that when you put the tip, the tip is actually putting the samples inside the wells. Okay, so because we are using the loading die, the samples will not float away. So if we don't use the loading die, they will just float everywhere. Now, because they are kind of bound to the glycerol, they will stay on the bottom of the well. Okay, so these are kind of loading die. So the agarose gel, because of the mesh-like structure, it will create a lot of burden for the DNA to move through. So if you look from the above, so let's say it's your gel with wells, and imagine it to be a mesh, so something like this, and the DNA should really put a lot of energy to move through. So in this case, the lighter, or the molecules with lower molecular weight, they will move faster. First of all, because they are smaller in size. Second, because it is easier to move something small because it requires less energy. So in this case, when we move through this gel, the lighter ones will run further than the heavier ones, and of course the heaviest ones will barely leave the well. And here you should know that the negative pole is usually colored black, while the positive pole usually colored red. So don't mix them up, because if you do the other way around, your DNA will move in the other direction. So it should move from minus to plus. So here we start the electrophoresis, and it's usually called gel run. So the procedure itself. Sometimes you might hear something like gel runs or just runs, and it means that we actually ran these gels. Okay, so what you do, you close the lid, and by closing it you connect this connector to your electrodes, and then through this connector it's connected to the power supply. So after you plug it, you will want to set up the power supply, and usually for DNA we set it up between 100 and 130 voltage. It should be constant, and for different sizes of the gels, of course, the running time will be different, but the general rule would be almost the same always. So you have a gel, and of course it has the length. So let's say it's 7 centimeters, and if you really want to see a good separation of the bands, you will want to run it so that the visible dye will travel not more than 2/3. So let's say it's 2/3, and your dye, the loading dye, should not move further than the 2/3. Why? Because whenever you have the small fragments of DNA, because they are very light and they don't require a lot of energy, they will move very fast. And just imagine that when you have this loading die coming to this end, you already lost the smallest fragments of DNA. So to prevent it, you would want to have this loading die move only to about 2/3 of the gel. When you really know that you're going to work with very small fragments of DNA, you would want to increase the percentage of the gel. So from 1 you can go up to 4. Of course, when you have 4% gel, it's very sticky, very solid from the beginning, so it might be very hard. I actually used the highest percentage I used was 3%, and it was already solid when I was pouring it inside the gel form. So in the end of this run, you will have nicely separated bands of your DNA, and you will see them as different pieces of fragments. Okay, so what are you going to do next? You're going to analyze your stained gels, and first of all, of course, you are going to determine the restriction fragment sizes. Then you're going to create the standard curve, and you're going to create it by using the DNA marker or DNA standard. In the lab, it's called Hint3. So this Hint3 digest is the DNA standard for this lab. Okay, so then you're going to measure the distance that the DNA traveled from the well. Okay, so you're going to start counting from here, so it's going to be a 0, and then count your band. So let's say you have 12 millimeters here, and you're going to write it down. Then you're going to determine the size of DNA fragments according to the DNA standard that you have. And after that, you're going to identify the related samples. So in our experiment, so we had 6 wells. So first well was loaded with Hint3 digest, then we had crime scene, then suspect 1, 2, 3 and 4. Okay, so here you can see the sizes of the pieces. So in total you will have 6 pieces of your DNA standard. So the heaviest one will be 23,000 base pairs, and it should travel the appropriate amount of centimeters. So let's say that it traveled 11 millimeters. Then the next one would be 9,400 base pairs, and it will go further for 2 millimeters, and so on and so on. So what you do, you determine your standard curve for the samples. So let's say that you have 3 bands, and you know that they traveled, so let's say first one traveled 20 millimeters, this one traveled 22, and the third one traveled 24. So in this case you could tell that, okay, so the third band is actually 2000 base pair long. But how do you determine the molecular weight of the second and the first bands? So what you do, you actually create the Hint3 digest graph. So you write it down like this. So for example, here you have the distance of 11 millimeters, and its molecular weight would be, so 10 here, then the next one would be 20, 30, 40, 50, etc. up to up to 100,000. So from here you take approximately 1/3 because it's 3,000. So between this 30,000 and this, a third would be around 3,000 base pairs, and you're going to draw it. Next you're going to take the 13 millimeters, which is here, and it is going to be 9,400. So what you actually do here, you identify 9,000, which is here, so it's 9,000, and between these two values you will draw the second point. Then you take the third bond from the hint 3, which is 15 millimeters. So you draw it on this 15 millimeter border, and its molecular weight is 6.5 thousand. So you identify the 6.5, which is 1, 2, 3, 4, 5, 6, and right in the middle between 6 and 7, which makes it roughly here. Then you take the 18 millimeters, and it's going to be on this line, and 18 millimeters is 4.4 thousand, and it is 2, 3, 4. 4.4 is somewhere here, which is this guy. Then you are taking 23 millimeters, and 23 is on this line. 23 is 2.3 thousand. 2 is here, and 3 is the third of the second section, so somewhere here. So this is 23, and finally we have 24 millimeters, which is 2000, so it's right on this line, so intersection of two lines. Okay, so this is your standard curve. So what you do next, so you already can say that the third bond of your sample is 2000 base pairs. So what you do next, you take 22 millimeters, which is 24, 23, 22, and you drop the perpendicular to your standard curve, and then drop the same on the y-axis. And in this case you identify it as more than 2,600. So let's say it's 2.8 thousandths. So for your second bond, the molecular weight would be 2.8 thousand base pairs. And finally you take the 20 millimeters, which is here, and when you drop it on the curve first and then on your y-axis, it's going to be about 3.4 thousand base pairs. So 3.4 times 10 to the power of 3 base pairs. So now you have identified the molecular weights for your sample. You're going to perform the same calculations for all of your bonds, and then compare them between, so that you identify the crime scene and your suspects. Of course, this is the exemplar values, so when you do your own calculations you will be using the photos of the gels, and of course the values of millimeters traveled from the well will be different. So let's say this will be 8 millimeters, this will be 11, etc., etc. The only thing that is constant here is these sizes. Okay, so the sizes of the hint3 DNA standard will tell you the approximate distance that these sizes could travel to, and according to this distance then you will be comparing your samples. So don't think that this distance is actually constant. No, the constant is this part, which is sizes. Okay, this is how you identify the samples and the sizes of fragments. And now let's talk about the plasmids. So what is the plasmid? It is the circular DNA usually found in bacteria, but scientists, they use it a lot in their practice. So if we have this plasmid and we insert some gene here, we can then transfect the cell with this plasmid, and this cell will produce this protein according to the gene sequence inserted in this plasmid. Okay, so this is a very powerful technique, and to use it you will need to know how the restriction happens so that you can calculate where to insert your gene, etc. So because it's circular, the number of fragments will be changing due to the number of restriction sites. So let's talk about these three plasmids. So the first one is being cut by the PST1 restrictase, and after cutting it actually produces three different fragments. One is 610 base pairs, another two are 305 base pairs each. So on your gel, if you take the well here, it will be looking like this. So you see only two bands because these two fragments are the same, but by knowing the actual construction or actual composition of this plasmid, you will be able to say that actually here in this band you have two separate parts of your plasmid. So next, let's say you have the plasmid 2 and we are going to cut it with eco R1. It creates three different bands, so three different fragments, sorry. So first one is 220 base pair long, the second one is 400 base pairs, and the third one is 620. So if you take the gel, so you have a well, then you have the first bond, which is 620, then you have 400, and finally you have the smallest one, which is 220. What if the plasmid actually has only one restriction site? So PST1 as the restrictase here, and we have only one restriction site. So on your gel you will see a single band, and in this case the plasmid is cut and it creates the linear DNA. So if you kind of rejoin it, it will create the same plasmid, but now after the cut of the restrictase we will have a single linear DNA molecule. Of course, if you know the fragment sizes, you can calculate the total size. So total here would be 220 plus 400 plus 620, which is 1240 base pairs. Here you would have 610, 305, 305, and it is 1210 base pairs. Here on this one you are not given at all, but the total would be the size of this bond. And actually if you have both linear and plasmid DNA loaded to this well, they will travel the same distance from the well because they are actually the same size. So let's say the total here would be 1200 base pairs. You could also have the representation of the task as the table. So let's say that you are given the plasmid cuts by the restrictases separately. So let's say that hint 3 cuts it to a single fragment of 1200 base pairs, which means that it creates a linear DNA, and the total of this Total of these plasmid would be 1200 base pairs. So it means that it only cuts once. For ECR1 we have two restriction sites, and they will look like this. For PST1 we have three restriction sites, and we obtain something like this. And we actually know that the total is the same for all of these drawings, because then we have the combined action of all three restrictases. Here, what we could obtain after we cut the restriction sites of this plasmid. So here you can see that hint 3 gives us a single piece of DNA, but it is linear. So if you take the PST1, it goes first to about a half, then 2/6 of the total, and then 1/6 of the total. ECR1 gives you a huge part of 800, and then a smaller one of 400. But we also know that we have smaller parts, so 10 is here, then we have 100 here, then 200 is actually from the PST, so here we would have 210. This guy would be the longest one, which is 390, and we are only left with this one, which is about 290 base pairs. So in this case you would be able to map your plasmid in a way that you can work with. And of course this is very rough representation of this plasmid mapping. So when you get to the lab where you perform this type of research, you will be given the sites like here. So let's say that we have the plasmid 5. You actually know that there are 3 different restrictases that could cut the plasmid 5, and you know that ECRI has 1 and 2 restriction sites, while the hint 3 gives you also 2. PST1 gives you 3 restriction sites, and by knowing the points on the plasmid, you can calculate the fragments and their sizes. So you know the total, which is 1440 base pairs, and you know that the restriction sites are at different places. So let's say this is 0. So first fragment is the sum of 150 and this small guy. So 1440 minus 1420 gives you 20 base pairs, and then you have these 150 and 0. So 150 minus 0, which is 150. So the first fragment between hint 3 and ECR1 is 170 base pair long. So this is first. So second would be 440 minus 150, and it gives you 290 base pair long. So third would be 619 minus 440, and it gives you 179 base pair long. So fourth fragment is 710 minus 619, and it gives you 191 base pair long. Fragment 5 is 1100 minus 710, which gives you 4390 base pair long. 6 fragment here gives you 1329 minus 1100, and it gives you 229 base pairs. Finally, you have the 7th fragment, which is 1420 minus 1329, and it gives you 191 again. So if you need to draw this gel here, so this is a well, then the biggest one is the 5th fragment, then we have the 2nd, so 290, 3rd, then we have 4th, 5th, and these two guys would be the same. Okay, so when you draw these gel, it will look like this: 1, 2, 3. So 1, 2, 3, 4 is like this. 5 is 70, so it's very close. Then we have 6, which is the same, so 91. Here we have 390, then 290, 229, 179, 170, and 91. So this is the representation of your plasmid digest after we run the gel. Okay, so this is the end of the today's lecture, and thank you all for listening.